In this first section, we're going to focus on the components of blood. So a couple of reminders. Blood is the only fluid connective tissue. So since it's a connective tissue, it has three basic parts. It has to have a fiber, has to have an extracellular matrix, and it has to have living cells. The formed elements are what we typically think of as the living cells, and then our plasma is what's known as the extracellular matrix. The fibers we don't really see until clotting starts to occur. Now, one other thing to note is that the pH has to stay between 7.35 and 7.45. Adjustments or modifications to this lead to dramatic changes and dramatic homeostatic mechanisms to maintain it. The other thing is the temperature of blood is a little bit different than the body temperature. This sort of makes sense when we think about its role in the maintenance or reduction of body temperature when we looked at vasodilation and vasoconstriction within the integumentary system. The last thing is to dispel a myth. Our blood is always red. It's simply a duller red or a scarlet red depending upon the presence of oxygen. So the first thing to look at is the blood plasma. Notice that blood plasma is 55% of the overall blood. It's mostly water. It has all kinds of stuff dissolved in it. The one I want to focus on real quick are some of the plasma proteins, primarily what we know as albumin. Now, albumin actually plays a huge role in osmotic pressure or osmotic regulation. So I want to remind you that osmosis is simply the movement of water, so an area of high concentration to an area of low concentration. Now, you should remember from biology, this is based on the relative ideas of hyper, hypo, and isotonic solutions. When you have a hypertonic solution, I have a lot of stuff that is dissolved, and so I actually have very little free water to move. If I have hypotonic, I have a lot of free water and very little stuff dissolved. So you tend to see movement of hypo to hyper. If we're talking osmosis and we're talking about the movement of water, well, let's think about our blood. If the blood is 55% of the blood and 90% of that is water, there's a, for lack of a better phrase, a vested interest in maintaining a certain amount of water in the blood. We have to have a certain amount of volume. Albumin is what maintains this osmotic pressure to ensure that more water stays within the blood than actually diffuses out of it by osmosis into our surrounding tissues. The formed elements are simply the living cells. So we're dealing with red blood cells, white blood cells, and then platelets, what we term cell fragments. Now, hematopoiesis is the primary function of the skeletal system because it only occurs in the red bone marrow. Remember that the red bone marrow is in our vertebrae, it's in the ilium of the coxa, and we see a little bit in the proximal ends of the femur. Now, every single blood cell, so every single formed element, red blood cells, white blood cells, and the platelets are formed from a single common stem cell, and that stem cell then differentiates into the different cells that we observe within our blood. Notice here that we actually have a primary division, so the hemocytoblast forms a lymphoid stem cell, forms lymphoid stem cells that then produce our lymphocytes, and then it forms what's known as the myeloid stem cells, which then produce the rest of our cells. So we have these various things. The main thing to notice here is the difference between the white blood cells and the red blood cells. Red blood cells, by definition, are anucleate. They have no nucleus. Their sole job is simply to form a reservoir or repository of oxygen. So they're geared and built primarily for that purpose. Whereas if you look at the lymphocytes or the leukocytes, the white blood cells, they have nuclei because they perform jobs that require them to make things, whether it be antibodies, histamines, things that are released in response to within the body in terms of defense, whether it be general or specific. Now, one of the things to notice here is how many red blood cells we have compared to white blood cells. That's a huge and astounding number when you think about it. Now, the other thing to recognize is the lifespan of an erythrocyte. Basically, it's 100 to 120 days. What's kind of cool is where it's eliminated. It's eliminated primarily within the spleen or the liver. What's cool about this is one of the things that you have to understand is there's an entire process associated with the breakdown of the red blood cell. So we want to take this little red blood cell and we want to salvage some of it. Ideally it's hemoglobin, so the heme unit, and then we want to get rid of the rest. And the rest of it is what actually is responsible for the color of our fecal matter and our urine. The fact that we have that slightly yellow and brownish appearance is a direct result of the red blood cells that we get rid of. The other thing is the red blood cells are what are the discharge, this breakdown of it is what's associated with the gall. So in our gallbladder, the release bile is a direct product of the removal and the breakdown of our red blood cells because our body is very efficient. We don't want to waste good working materials. Now, the rate is controlled by a hormone produced by the kidneys, and this is known as erythropoietin. Now, notice what causes the change in our erythropoietin levels. It's literally a direct result of the oxygen levels in the blood. Think about if you moved here from, say, somewhere like Florida or California where you're at sea level. We are about 4,000 feet. That's actually a fairly significant change in altitude, and it tends to mean that those people will tend to be out of breath more readily, they fatigue easier because their body's not used to carrying enough oxygen via their blood. The same thing will happen to you if you ended up going to Denver. So when you talk to or hear about the different athletes like the bikers, the Tour de France bikers and things like that, they tend to train at very, very high altitudes. For one reason is to get enough red blood cells to be able to carry adequate amounts of oxygen as they go up and down through that altitude. That lack of oxygen causes the production of the erythropoietin, which triggers the red blood cells, or triggers the stem cells within the red marrow to produce blood cells. Now, the hemoglobin is huge, as we've already talked about, because it's the hemoglobin, the presence of hemoglobin, that determines how much oxygen we can carry. The number of red blood cells are then also impacted by that. So the main thing here is that look at the number. We have roughly 250 million hemoglobin molecules. Now these are molecules, remember we're talking proteins, per red blood cell. Remember that the red blood cells outnumber the white blood cells roughly 1,000 to 1. So I have 250 million on a single cell. Remember that also that our red blood cell was basically a tiny cell. It's one of the smallest cells in our body, and it has 250 molecules within it strictly for the purpose of carrying oxygen. Each hemoglobin molecule can bind with 4 O2s. So that means I have 4 times 250 million. So that's 1 trillion oxygen molecules, O2s, per red blood cell carried throughout your body. And we have a lot of blood in our body. Remember, roughly 4 to 5 pints. It's a lot of cells carrying a lot of oxygen. And again, this is crucial to homeostasis because every single cell in our body requires oxygen. If we have one cell die, that could potentially impact an entire area of tissue. If it impacts an entire area of tissue, it could potentially cause problems within the organ. If the organ starts to fail, then we have an impact on the organ system. If the organ system starts to malfunction, that can affect the overall organism. So it doesn't take much if I have just a single cell die, a single cell, to trigger a massive cascade event that could potentially lead to death for the overall person. Now, quickly, white blood cells are our defense against disease. These are generally going to produce specific and general defenses. The one type of cell, the macrophages generally, are what can move in and out of the blood vessels. We see them kind of the amoeboid idea. They are what seem to respond to the engulfing, so they're responsible for the phagocytosis within our body to get rid of specific foreign invaders. There are normal is 4 to 11,000. Notice that. If we have abnormal leukocyte levels, this is sometimes associated with different drugs and or infections. So your body naturally has more white blood cells when you are fighting an infection. Here's the breakdown associated with the types of leukocytes. So we have the granules and the granulocytes versus the agranulocytes. In essence, they're responsible for releasing different granules associated with our general defenses. Different ones have different roles within that overall function. It is our granulocytes that are also responsible for our allergic responses and the release of histamines. The different cells have a role in everything from general defenses, inflammation, etc. Now the agranulocytes are basically what we tend to think of as our antibody-producing cells. They look a little different. They have a very, very large nucleus, which kind of makes sense if they're producing antibodies. And they again also play a different role, but still a role in terms of maintaining the defense of the body against foreign invaders and foreign attackers. The platelets ultimately are simply, they look like cell fragments, cell parts, but we have and use them for the clotting process simply because it's the platelets that get wrapped up in a scab. So when you look at a scab, you're actually looking at dried up platelets.